Dear students, today I am going to discuss about two topics. One is catalytic antibodies or CATABs, and the second one is generation of immunoglobulin gene libraries. So first I will start with catalytic antibody. So if I go to the history in brief about the production of and discovery of catalytic antibodies. So first, this particular antibodies or the concept of catalytic antibody has been suggested first by William P. Jenks in 1969, and the possibility of catalyzing a reaction by the means of using an antibody that binds to the transition state was first suggested by William Jenks. Later in 1994, the other two scientists, Peter Soos and Richard Lerner, they have developed the catalytic antibodies, and they have utilized the name of catalyzing monoclonal antibodies to Catmab or catalytic antibodies. Later, the pioneering work done by Dr. Sudhir Paul, who publishes a paper in 2008 giving the first example of hydrolysis of HIV protein GB120 by using an APSA. And this, through this discovery, Dr. Paul has revolutionized the application of ABZYME in different disease treatment. It may be viral or bacterial infections. So the catalytic antibodies, which are also known as ABZYME, or else known as Catmab, are most often called as catalytic antibody of a monoclonal antibody with catalytic activity. It's a single molecule of an antigen of an antibody enzyme complex which is capable of catalyzing the destruction of thousands of target molecules. And on the basis of differences between antibodies and enzymes, is that the former means the antibodies defines the complementary structure is the antigen in its ground state, while the enzymes they bound the specific substrate at their high energy state. So ABZYMEs are usually raised in animals, laboratory animals, artificially by immunizing laboratory animals against a synthetic organic molecule known as hapten. But some natural ABZYMEs can also be found mostly in autoimmune disorders in human. So ABZYMEs bind strongly to the transition state with high association constant and enhancing the reaction rate by reducing rotational entropy of the transition state molecule. So the name ABZYME was proposed only because of this AB comes from antibody, enzymes from enzymes. So altogether they are termed as ABZYMEs. They are actually the catalytic monoclonal antibodies. Now, what are the sources of ABZYMEs? So ABZYME source can be of two types. One is artificial source and another one is a natural source. So when we are talking about the artificial source, so that means there we have to develop it. Means we have to inject a hapten either in laboratory animal or in human, and then it will develop the antibody that have the catalytic activity. Or else we can obtain catalytic antibodies or ABZYMEs from human or animal, those from human or animal serum, either by developing artificially, or maybe the patient or the human being that are suffering from autoimmune disorders. So these are the enzymes that are capable of hydrolyzing proteins, nucleic acids in the form of DNA or RNA, as well as polysaccharides. So when these enzymes, naturally occurring enzymes, they have proteolytic activity or protein degrading function, they are called as proteazymes. And they specifically hydrolyze a specific protein in patients with autoimmune diseases such as in bronchial asthma, in multiple sclerosis. So these are the autoimmune disorders. There, this particular proteazyme can be found. And in case of DNA hydrolyzing activity, they are known as DNA enzymes. So the pathogenic role of DNA enzymes is still unclear to us. However, these DNA enzymes they are acting as a powerful regulator in our process in cancer treatment. But how? What is the molecular basis? What is the mode of action? Is still unclear to us. Now I'm coming to the principle. So the production of enzyme is based on two following principles. One is enzymes act by binding the transition state of a reactant better than the ground state. And the second one is that the antibodies which bind to specific small molecule can be produced by coupling this small molecule to a protein carrier and using this protein for immunizing experimental animals. So this is the principle of production of monoclonal antibody having catalytic activity, that is Catmab or catalytic antibodies. So catalytic antibody functions is an enzymatic function, and the mechanism of action and mode of action of this enzymatic function of Catmab is by lowering the activation energy of the transition state of a chemical reaction or the chemical reaction, thereby enabling the formation of an otherwise less favorable molecular intermediates between the reactants and the products. So if you look into the picture, here you will see this is a simple enzymatic reaction or non-enzymatic reaction profile. So along y-axis we are showing free energy change, and along x-axis is the reaction coordinate or how long it will take to carry on the reaction. Now, if this red line that is showing enzyme, that is showing chemical reaction or biochemical reaction that is undergoing without the association of any enzyme, and the blue line is showing biochemical reaction or any chemical reaction that is taking place in presence of a specific enzyme. So, if you look into this picture, you will clearly see that at their high energy state, that is known as the transition state, so energy differences is there. So here in the reaction that is not involving enzymes, or the reaction that is involving a particular enzyme, there is a difference between the activation energy. So this activation energy difference is actually due to the association of enzyme. And in the chemical reaction where enzyme is taking place, the activation energy or free energy is lower in case of the reaction that is undergoing under the presence of or under the function of enzyme. So here, if an antibody we can develop that will bind to a specific molecule that is structurally and electronically similar to the transition state of a given chemical reaction, and the developed antibody will bind to this and stabilize the transition state just like a natural enzyme by lowering the activation energy, by reducing the rotational entropy of a particular reaction and thus catalyzing the reaction. So this type of enzymes, monoclonal antibody that has a catalyzing function are known as Catmab. So by raising an antibody to bind to a stable transition state analog, a new and unique type of enzyme is produced, and these are known as enzymes or Catmab. So this is the scheme how they are actually functioning in our body. So if we are developing it using using haptane along with the antigen, so the antigen, that is the transition analog that acts as an antigen. So this triangle is showing this is the transition analog, and this is the substrate, that is the haptane, and we are injecting both into mice blood, and when it is mixing into the mice blood, there is a transition state, and they will develop the mice blood. B lymphocytes are present, they will produce their complementary to the transition state, means complementary to antigen, that is the antibody, and now the antibody will conjugate with the antigen, the transition state analog, and will give rise the product. So here, this is the abside they are preparing. Now, what type of reactions can be catalyzed by enzymes? So there are 8 to 9 different types of reactions that can be catalyzed by enzymes. Are amide hydrolysis, transesterification, photolytic cleavage, photocleavage, photodimerization, decarboxylation, oxidation, cyclization, then reduction of diketones, hydrolysis of enol ethers, like that. So these are the reactions that can be catalyzed by enzymes. Now, where we can apply these particular monoclonal antibodies? So for the treatment of cancer, for the treatment of HIV, drug detoxification, and also against weight gaining, and antibody-directed enzymes in product therapy, in treating allergy, treating bacterial infections. So these are the field where we can apply enzymes for mostly as a therapeutic, have a therapeutic value. So as I said that we can apply it for HIV treatment, and pioneering work done by Dr. Sudhir Paul, that it can be an agent, it can be a means for the prevention of or the potential HIV treatment. So in June 2008, the issue of journal Autoimmunity Review, published by researchers of Oxford University of Texas Medical School at Houston, they have announced that they have engineered an enzyme that can degrades the superantigen region of the GP120 of CD4 binding site of HIV. Because this particular protein GP120 that is necessary for HIV to attach, to bind with the CD4 positive T cell, and it doesn't chain across different strains, and is a point of vulnerability across the entire range of HIV variant population. So they have proposed that the enzyme does more than bind to the site, it catalytically destroys the site, rendering the virus inert, and then can attack the HIV virus. So this type of chemically synthesized enzyme can be functional against HIV treatment. And it has been observed that a single molecule of enzyme can destroy thousands of HIV, thousands of human immunodeficiency virus. But this is no doubt this is the advantage of catalytic antibodies. But catalytic antibodies have some disadvantages, and the disadvantages are they have low affinity, they are highly expensive, is high production cost, slow turnover state, and also empirical production. So these are the disadvantages of catalytic antibodies. Now I'm coming to a different topic, that is generation of immunoglobulin gene libraries. So various methods have been employed to clone the genetic diversity of antibody collections. And after isolation of mRNA from B lymphocytes and preparation of complementary DNA or cDNA, the construction of immune libraries is usually done by a two-step cloning or assembly procedure. So very large single pot NAB antibody gene libraries are generally constructed by two or three separate cloning steps. So here in the two-step cloning strategy, the amplified collection of light chain genes is cloned into the first display. So here in the first display vector, first we are cloning it, and since the heavy chain contributes more to diversity because of its high variable CDRH3, the heavy chain gene collection is cloned in the second step into the plasmids containing the light chain gene collection. So in this step, in this three-step cloning strategy, separate heavy and light chain libraries are engineered. So here you will see that since the libraries with more than 1,010 independent clones have also been accomplished by conventional transformation, most of these complicated methods are rendered unnecessary, particularly as they may result in decreased genetic stability. So in summary, we can say that antibodies can be selected from either type of library, naive or synthetic, if the assembly by cloning or PCR and preservation of molecular complexity is carefully controlled at every step of its construction. Libraries of more than 1,010 independent clones can be generated. And a method can be proposed like that. The methods describe the generation of human naive or immune SC FB fuzz antibody gene library. So here, this antibody gene library is by a two-step cloning strategy already proven for different naive and immune libraries. So in the first step, the lymphocytes will be prepared here, this blood sample. The lymphocytes will be prepared for mRNA or total RNA isolation. The isolated mRNA will be then reverse transcribed to prepare the cDNA. And a set of human antibody gene oligonucleotide primers will be used for the amplification of both the variable heavy chain as well as the light chains by PCR for the amplification purpose. And in the second PCR, restriction sites for the cloning in PHAL14 will be added here. So the cloning will be performed in two steps. First, in the variable light chain will be cloned, followed by cloning of the variable light chain, then followed by cloning of the variable heavy chain. In the end, the antibody gene libraries will be packed using a helper fuzz and analyzed by immunoblot. So the scheme of the total procedure, what I have shown here. So this is the packaging of a library, and this is the fuzz, SCFD fuzz. So that is how we can develop, we can generate immunoglobulin gene libraries. That's all.